Traceability, for having me here this evening. It's really hard for me to believe the words I'm reading on the screen, that we are looking, it is just 1 month under a year from when this all began here, at least in New York. And the fact that we are talking about vaccines, that people are already getting vaccinated, is just quite amazing to me. So I wanted to start out this evening with a quote from one of the, let's see if I can, one of the famous scientists in vaccinology, Louis Pasteur. Fortune favors the prepared mind. So we're here tonight to learn a little bit about vaccines. I always have the disclaimer of, I'm not a medical doctor. I'm a PhD in molecular biology, as Swadesh mentioned. And while I have spent my career designing tests, we often work hand in hand with companies that make vaccines, because part of what you have to test for once somebody's vaccinated is that they produced an immune response, and you test for the antibodies. And that's one of the things we do at Siemens. So I just wanted to start tonight's discussion out with a reminder that, when you look at all of the achievements of health, not only in the United States, but really around the world, one of the top 10 greatest achievements, and probably, in the top 3, are vaccines. It wasn't that long ago. I can remember as a kid, I have had many of the diseases that, children never get anymore, because at the beginning of the 20th century, there were just a lot of infectious diseases that were widely prevalent in our country. For example, in the 1900s, there were 21,000 smallpox cases, 469,000 measles cases, and we would have, roughly 7,500 people dying a year from them. So by the time vaccines were introduced, and mostly through childhood vaccinations and through school programs tied to forcing people to get vaccines before they attended school, a lot of people died and got very sick from the diseases that were around. So based on the success of those, moving into the 21st century, new kinds of vaccines were used, and I'll talk a little bit about that evolution. Now we routinely vaccinate kids for meningococcal and pneumococcal diseases, rotavirus, which is a disease that affects small babies, and we even have a vaccine that prevents cancer, and that's against human papillomavirus, which is the virus that causes cervical cancer. So that, in addition to all of the vaccines we get as children, have really improved health. Just as a reminder, I wanted to show this slide. I know it's a little bit intense, but to remind people what these diseases look like. So right here is a child with chickenpox, here a child with smallpox, a child with mumps, a child with measles, diphtheria, and polio. So these were very severe diseases. An example of kind of the power of vaccines is shown here, where polio has been eliminated in the United States. And if you look at this graph here, which has the decade, the 1950s, 60s, and 70s, against the cases of paralysis caused by polio, you can see in the 50s, before we started vaccinating children, there were about 15,000 cases a year. And I think those of us who are in our 50s and 60s probably know 1 or 2 people who actually had polio and may still have paralysis in parts of their body because of that. But by the 1960s, we had a very successful childhood vaccination program, and we were down to 100 cases. So that's just an incredible drop. By the 70s, there was about 10 cases per year. And by 1979, we had eradicated polio from the United States. A very similar story in the world. An effort was led by the World Health Organization in the 1990s. You can see here in this graph, all of the pink countries highlighted, or continents really, were places where polio existed in 1988. And worldwide effort went on. Now polio exists in only 2 very small places, one at the border of Pakistan and India, and in 1 country in Africa. So again, an amazing story. And actually in 1979, smallpox was eradicated from the globe. So what do vaccines do? Vaccines really are made to do the same thing that an infection does, only not get you sick. So what happens when your body first encounters a virus, which is shown here by this green little particle, your body's first responds by producing a kind of antibody called an IgM antibody. And if you look down here at this graph, the first time a human sees a pathogen, they react and produce antibodies at a very low level. So your body is just basically learning, it's understanding what the pathogen is and what it needs to react. And on the second exposure to a virus, now your body knows that your immune cells have a memory to them, and they can remake the antibodies against the virus, and they can do it in a second very powerful response. So what we're trying to do with vaccines is create this first step, this ability for your body to recognize something that is foreign. And then the next time you encounter it, you'll be able to have a really significant response. So these are the types of vaccines that are available now. There's, vaccination, the practice of it has been around for a very long time. In the 17th century, Buddhist monks used to vaccinate each other, from their sores from smallpox, similar to what was done in the West by Edward Jenner when he first was experimenting on the smallpox vaccine. So really the first vaccines were live, actual virus transmitted from one person to another in a very small dose, so that hopefully they didn't get the full-blown infection. But that was a very, very, very risky procedure. So by the time in the 19th century we started to make vaccines, they were they were derived from live virus, but they were put through a process that would attenuate them. And what that means is it would keep them from being highly infectious. So and so that they would not go on to cause disease. And our current measles, mumps, and rubella vaccines, the chickenpox vaccine, are these kind of vaccines where they're attenuated. We also have inactivated vaccines that are derived from virus, but totally inactivated. Currently, the polio vaccine we used is an inactivated virus. We used to, in the 60s, have a live version of the polio vaccine. That's actually the one I had, but it carried a lot of risk with it, and people would actually get polio from it, so they stopped producing that. So these were really what carried us through the better half of 19th and 20th century. And then along in the 1980s, we started to begin to develop vaccines that where we used another virus as the carrier of the vaccine. So this is where genetic engineering began and protein engineering with some of these viruses. So what happens is you take a disease-causing virus and you combine it with a low pathogenic virus, so something like a common cold. So you actually use the shell of a common cold, and then you take the material that encodes all of the harmful viral proteins and put that into a harmless shell. And you can actually create this vaccine, which when injected into people can replicate and then trigger your body to respond to it. This is not used very much in human medicine, but it is in veterinary medicine. What's used right now in human medicine a lot is the same similar approach. However, this virus cannot replicate. So you'll create an engineered virus that will be able to go into a host and produce an immune response, but it won't create another round of infection. There's currently an Ebola vaccine that is produced in that kind of a vector. The DNA and RNA vectors are nucleic acid vaccines, and I'm going to talk in detail about them because that's really what's available in the United States right now, probably what some people on this call have actually had. And those vaccines are derived of nucleic acid. And so nucleic acid is the chemical that actually carries all of the heritable material in humans and in viruses. So you can actually code a piece of DNA or a piece of RNA to be able to go into a human cell, use the human cell machinery, and start to produce viral proteins. So that's been the strategy, the RNA vaccine strategy for COVID. And then the last strategy seen all the way at the right-hand side of the screen are genetic engineered particles, where you can make a particle that has on the outside of the structure of it these little knobs, which is actually what your body sees as foreign, but it doesn't have anything inside of it. So again, it's non-infectious, but it is strong enough to elicit an immune response from a human. So another thing that you hear about a lot when we talk about vaccines is herd immunity, and really from a public health perspective, this is what we're trying to achieve. So if we look here at the top of this cartoon, when no one has immunity, which is where we were at the beginning of COVID, if one person gets infected, they can just spread that very rapidly, especially in the case of a highly infectious virus like COVID-19. Once people get immune to it, and you start to have this inner circle here shown by the little men in green who are immune and the people in blue are not, if someone gets infected, they have less ability to transmit the disease because there are just fewer people who are susceptible to it. And the goal of vaccination is really shown at the bottom, where you want to have enough of the population so that it's unlikely for an infected person to come in contact with an uninfected person. And in this kind of a situation, you can actually have some people who are not vaccinated who remain protected because the majority of the herd is providing a wall of protection. So it's really a numbers game here. So when scientists they have to look at how easily transmitted a virus is, and that's how they make a calculation of how much of the population has to be vaccinated to get into this herd immunity state. So I want to talk specifically about how RNA vaccines work. So they work based on the principle of what we call the central dogma of molecular biology. And basically what that is what we know right now about the way we replicate our cells is all of our genetic heritable information is stored in the cell nucleus. So we're looking at one single cell here, a eukaryotic cell, and humans are eukaryotes. And in the nucleus of a cell, which is kind of like the brains of the cell, is where DNA lives, and all of the instructions for every, in the case of a human, for how to make a human are within that molecule, that chemical of DNA. DNA exists in a double-stranded form, and this and a couple other things about the chemical make it a very stable chemical. So this is the basis of things like why, Jurassic Park, you could find old dinosaur bones and get DNA out of it. It's very stable. It hangs around through heat, through rain, through cold. But the first step in turning the DNA information into a protein, which is the last step here, is there's an intermediate step, and that's where the chemical RNA is involved. So like DNA, it's a nucleic acid, but it is much less stable. So when you hear about the RNA vaccines being not very stable, it's because this chemical form is not nearly as protected as DNA. And that is a bit due to the fact that its purpose needs it to be very transient. We don't want RNA to hang around very long. So what happens is DNA is transcribed by enzymes in the body into RNA, And this RNA message is a faithful copy of this DNA. The message then leaves the nucleus of the cell, goes into the cytoplasm of the cell, where there's the machinery that's necessary to create proteins. And so the RNA carries a signal, and a protein can be made from that signal. And what happens with viruses is they really come in and take over this machinery when they infect a cell. So viruses are either composed of RNA or DNA, and they basically just put themselves into this system and take it over and make us sick. So when I think about that system, I like to think about it like this, with this kind of an analogy. So I look at DNA as a blueprint, right? It's kind of like a, tells you how to build a human or a flower or a bee or whatever the DNA is resident in. And messenger RNA is kind of like a construction worker or an engineer, and they're able to translate what's in the DNA into a message which is built into a building. So for the SARS-CoV-2 virus, which is the causative agent of COVID-19, this is a cartoon of what the virus looks like when you look at it under a microscope. And these knobs here are really what comes into contact and allows the virus to infect a human cell. So these are also what we want to block with an antibody response so that the virus can't get into our cell. So that's what the outside of the virus looks like, and it actually acquires this green part of its shell as it buds through the human cell that it's infected in. So as the proteins for the virus are built, they are pushed through the cell, and that causes another round of infection. Inside the virus are the RNA, which gives the instructions for the virus protein. And that's what scientists have used to create like a fake infection with a vaccine. So in this cartoon here, we can see that what scientists did with the RNA vaccine is they didn't take all the information that was needed to create a whole COVID or SARS-CoV-2 viral particle. They just took the sequence of the RNA that gives the instructions to make this protein. And that's the protein that has to be blocked to make sure that it doesn't infect cells. So scientists made a synthetic version of that. They packaged it in a way that the body could take it in, and it could, once it's injected, it can go into human cells. And then our human cells would actually start to produce viral protein. That protein would be pushed outside of our cells, and our immune system recognizes it as foreign and starts to produce antibodies against it. And so that's the basis of the RNA vaccine. So vaccines carry side effects, and they always do. Actually, every drug, every medicine, there's no medicine that is without side effects. And the nature of the side effects really doesn't come so much necessarily from the bacteria or virus or toxin anymore. A lot of it comes from the formulation of the vaccine. And really what I'm talking about here are the severe side effects, right? So when you get a vaccine, you're actually creating your body, an immune response that it would give to an infection. So you feel not very good, right? You feel like an infection's coming on because your immune system is working. But the severe reactions that some people experience with vaccines really come from an allergic reaction to the formulation of the vaccine. So sometimes there are salts or preservatives or nanoparticles if a virus is housed in one of them, and they can sometimes trigger allergic reactions. So there are some other myths of vaccine that I just wanted to address early on. Some people say that natural immunity is better than vaccine-acquired immunity. So at the beginning of COVID, there was talk of just letting everybody get infected naturally and produce a herd immunity. So that's not true. A vaccination, the immune response is the same no matter if it's elicited against a virus or part of that virus. and it's also, prevents a lot of disease, as we talked about at the first couple slides. People also say that vaccines contain unsafe toxins. So at one time, early on with vaccines, there were some really, some caustic kind of chemicals that were tried out as formulations, and that's where that comes from. Things like formaldehyde, mercury, or aluminum salts. And so these have all been banned from vaccines. So the vaccines that we get now don't have those kind of materials in them anymore. The other thing I wanted to talk about, because you'll hear a lot in the news, is why we fear the medical establishment. So I have a couple of examples here, but unfortunately, to be really honest, there's a lot of examples of pretty horrible things that we did under government sanction in our past, and really up until the 70s. So one thing that people talk about in particular with the Black and Brown community is the 1930s Tuskegee study of untreated syphilis, which was a really horrible government study where people were being looked at and experimented on and not actually given the information of what they were participating in, and it went on for 40 years. There were cases, in the 50s and the 60s of experimentation being done on hospitals that in particular had poor people in them. And I even threw this one example in here in the 1960s where the Army released a bacillus, which is a bacteria that produces toxin into the New York City subway system. So, there was a time in our history where things were not really well controlled. So since the 1970s, there are a huge body of law that has been put into place to prevent those things from happening. Starting really with the Federal Food, Drug, and Cosmetic Act, which was started in 1938, really against people who would try to sell elixirs and things like that, from carts in the street. And actually, it's now been revised to include a lot of the protections that we now have in place during clinical trials to make sure that patients are aware of what's happening to them and they get all of the information and they have informed consent. The Kavauer-Harris amendments of 1962, which were inspired by the thalidomide issue, which was used with pregnant women with morning sickness and created mutations in babies, was pulled off of the market, was actually pulled off of the market in Europe. So the FDA actually prevented thalidomide from getting to the United States. So it was only seen in Europe. And then, another series of protections were put in place because of what we saw happen outside of the United States. And then in the 1970s, there was the Medical Device Amendment Acts, which put into place a lot of law to protect around injuries. And what the FDA has to do now is once a medical product is released into the market, including vaccines, they have to be followed for their life. Our company does that too on medical devices. So once a medical device or a medicine or a therapeutic or an implantable is put into the market, companies who manufacture them are required to follow the people that are being using the product and monitor adverse events and report them back to the FDA, and the FDA can take action if they feel necessary. And finally, as early as 2005, a lot of patient safety and quality was put into place by an act in 2005. And this is where a lot of the HIPAA law comes into place. So when you're going to your doctor and they make you sign all those papers to make sure that your privacy is protected. So there's a lot of laws that have been put in place now. In addition to at the federal level, a lot of states, including New York State, has its own rules and regulations regarding patient safety. So you have to pass through, and New York is a very rigorous state in that regard. There are two states in the United States, really California and New York, that have very high standards regarding medical practice. So I listed here, and you'll get a copy of this presentation where you can find the medical laws for the state of New York. So in particular, the laws that really protect people who enter into clinical trials, which is what we need to do to prove that something is effective and safe before it can be made commercially available, are two really important things that were put into place by law. One is the practice of what's called an institutional review board. So this is a review board that is independent of the group that is conducting a clinical trial. So when you want to do a clinical trial on anything in the United States, you have to prepare all of the documentation about expectations, the protocol, what's going to happen to the patients, how they're going to be followed, how all the data are going to be handled, and a panel of experts has to review it and assure safety for the people who participate in a clinical trial. Part of what the Institutional Review Board reviews is the informed consent form, which would be given to every person who engages in a clinical trial. And in that informed consent, it has to specify what exactly is going to happen to them, if they're going to be given a treatment, and what they would do if they wanted to get data for that, or if they had to report any kind of sign or symptom based on what they're experiencing during the clinical trial. An office that has oversight to that is the Office of Human Research and Protections. So they are the main keeper of all of the regulations around clinical trials. So just to give you an idea of what informed consent looks like, you have to be able to tell an individual who's going to be in a clinical trial the purpose of the research, how long it's going to take, what exactly the study involves, that the research participation is voluntary, and that they can stop no matter what at any time they want if they feel they're not receiving the care they need. They have to be able to tell all the procedures that will be completed during the clinical trial, if there are possible benefits, if there are risks, any possible discomfort that they might feel based on early trials that have been done, any alternative procedures that might benefit the person who's a subject of the research. We have to be able to explain how the subject's information will be kept private during the entire process, including for the duration of how long the data is going to be held. Whether there's any compensation for the medical treatment or just for enrolling in the study. Then finally, the subject's rights. And again, if they're able to refuse to pull themselves out of the trial at any time. And then contact information so that they can report any kind of injury or concern that they have during a clinical trial. So this is a very different process than we used before the 1970s. So when you're looking at clinical trial design, there are a couple of really important things that you want to do. So one is control the study. So one way that we control the study is by using something that's called a placebo. So you'll hear that word a lot. So in the case of a vaccine trial, and the ones that were used for the mRNA vaccines that are now being given to people, that clinical trial had an arm, as we refer to it, that where people were given a placebo. So there were a group in that trial that weren't given a vaccine. They were given a shot, but it wasn't of a vaccine. It was of something inert, like a saline solution. And we do that because we need to have a group of people who are representative of the normal untreated population so that we can really understand if we see an effect of the vaccine that we're trying to test. So that's one thing that was done in the vaccine trials. Another concern in a trial is making sure that you eliminate as much bias as possible. So one way to do that is through randomization. So when you have people sign up for a clinical trial, for instance, a lot of times, whether they're assigned to a vaccine or a placebo arm, is done randomly by computer assignment. So sometimes if you have humans doing this, for various reasons, they might put one person in one group and one person in another. So a lot of times we'll use computers to help guide how this clinical study is carried out. Another thing that we do to eliminate bias is blind some of the participants in the trial. And you can have a single or a double or a triple blinded study, and there are actually other ways of blinding. But when we say blind, that means in the case of a single blinded design study, the subject or the person who's receiving the treatment here, they don't know what they're getting. They don't know if they're in the vaccine arm or in the placebo arm. You can also blind the investigators who are part of the treatment and administering that vaccine. So they don't know if they're giving a real vaccine or a placebo to the recipient. And then finally, you can also blind the people who are running the clinical trial overall and looking at the initial data as it comes in. So those are the concerns and some of the things that we put into a trial design to make sure that we are getting the best kind of data that we can. So a clinical trial, a usual clinical trial goes through several phases and in general 4 phases. So this is really after research and research is done, and I'll talk about that in a minute. But a basic clinical trial is once something is demonstrated as completed development, first you usually engage in a small trial of about 20 to 100 healthy volunteers. And really the first question people want some assurance on is it safe? Is what you're injecting into somebody going to cause a severe adverse reaction? So they're looking for serious side effects. And they're looking for the beginnings of, will the vaccine be effective? Will you generate an immune response? If you pass through that stage, then the next phase will include several hundred volunteers. And now you're really starting to try to ask questions around short-term side effects, starting to really characterize a bit more the body's ability to mount an immune response, and starting to look if the vaccine is actually protective. Phase 3 clinical trials usually have 1,000 or more, and this number is really dictated by a lot of statistical considerations. And now you are looking at disease rates against different types of people in your group, and I'll show you a little of the clinical trial data, which shows how they look at race and ethnicity and ages, people of different ages. And now you're really focusing in on, will the vaccine be effective? So we often call that efficacy. So vaccines are really tested for 2 things, safety and efficacy. And then the final phase is after approval, but the study doesn't stop there because it's required by law, as I said, to monitor all therapeutics once they're out on the market. So we'll continue to collect data. So one of the things people had mentioned about DNA and RNA vaccines is that they're new. So, there's part of it that's true. They have, until now, there were no commercial DNA or RNA based vaccines in the United States. So from a vaccine technology, they are new. However, I'm showing here in this cartoon a typical timeline for vaccine development. So you can see it takes roughly 5 to 10 years to get something from the very beginning of its research exploratory phase, and then into preclinical trials, into clinical trials, and through FDA approval. So RNA vaccines have really been in this phase for 10 years. So that's a pretty long time. So from that perspective, they're not new. So as I said, this is a typical traditional product timeline, and you can see it's done in a very linear fashion where you do one step, then you move on to the next, then you move on to the next. But what we do in a pandemic situation with what's called an emergency use authorization is we try to speed up this timeline. At the same time, you want to speed up the timeline, you don't want to remove any steps. So this is how they do it. They keep all of the steps. They have to pass all of the steps any regular vaccine would go through, but they do things like the FDA starts the review during the clinical trial. Instead of waiting for the whole thing to be done, all the data to be analyzed and wrapped up tight, the FDA starts reviewing it at a very early stage, and the review continues to roll. So as data comes out, the FDA is reviewing it, so it's not all left to the end. In addition, another part that takes a long time is taking a very small scale of a vaccine that was produced for these clinical trials and taking that to a manufacturing plant and making enough to vaccinate a country or the world. So the manufacturing process is started much earlier. One of the things that made this process very rapid for the RNA vaccines is from a manufacturing standpoint, they are much easier to make. So all of that contributed to the fact that, by the end of December, we had approved vaccines in the United States. So here's a look at the trials that were conducted. On this side of the screen, the Pfizer trial, which had 45,302 people enrolled. The Moderna trial had 30,000 people enrolled. And here's kind of what that looks like. There's many clinical trial sites, so not just one or two. Here was 150, in the Moderna 89. The majority of them were done in the United States, but when these companies were anticipating their vaccines would be used across the globe, so they wanted to have clinical trial sites that represented people all over the world. You also really want to pay attention to racial and ethnic distribution. You want that to somewhat, reflect the population that you're going to be using something on, and also, paying attention to ages. So about 40% of the people in the Pfizer trial were between the ages of 56 and 85, and 25% were above 65 for the Moderna. I believe the Moderna one is now approved for people 16 and up, and Pfizer for 18 and up. And the reason for that is, you usually don't start out focusing on children unless it's a vaccine that's supposed to be used exclusively for children, because those trials, are yet a whole nother level of work required, because children's immune systems are significantly different than an adult. So, you can see there's a lot of people that go into the studies. So this is a table from some of the results that came from the Moderna vaccine study, and this is looking at efficacy. So what they're looking at here were, the placebo arm. So those are the people who did not get the vaccine against the group of people that got the vaccine. And of the vaccine recipients, these were only those that showed symptoms. So right here, people who got the placebo but actually showed symptoms that were known to be caused by COVID, and the same thing with vaccine recipients. So this is all of the people who got sick after they got vaccinated. So you can clearly see that in the placebo arm, there were lots of people who got sick. When you compare the people who had the vaccine, you can see, it's dramatically decreased, sometimes, going from hundreds to single digits. So you can see immediately there's definitely an effect of the vaccine. And the thing that was so exciting about these vaccines is when you looked into the people who got severe cases of COVID. So again, the placebo data is shown here, and you can see those people that got the vaccine shown in this column, there were no cases of severe disease. So while the protection might not be 100% of the vaccine, it did the job of making sure that people did not get severely sick and die. So that's an important piece to remember about these vaccines. And it did dramatically decrease the amount of people who got sick at all. So the Pfizer data is in this slide, and they decided to present it a little bit differently. I really liked how Moderna showed all of the different symptoms that people got. Here, Pfizer just kind of lumped people into people who had, severe COVID after the first dose, between the first and second dose, after the second dose, and then greater than 7 days after the second dose. So here are the people who got the vaccine. Here are the people who got the placebo. And you can see, roughly here there were 18 cases compared to 2 severe cases of people who got vaccinated. So I also think this is a good example of how different clinical trial data can look, even though the outcome is basically the same. The virus offers protection from severe infection. So the other aspect of the trial that we look at is safety. And that's shown here for the Pfizer vaccine. And basically what they're looking at is any kind of what's called an adverse event. So not getting sick with COVID, but if you had any kind of a reaction, mostly an allergic type reaction, and it's graded whether it is likely related to the vaccine or some other event that happened to the patient as they were going through the trial, whether it was severe or life-threatening. And you can see here again, people who were in the placebo arm had a certain rate of adverse events compared to people who took the vaccine. So again, you're triggering in these people who took the vaccine an immune response, and there is an effect of that, and people do, report symptoms from that. What we're looking at here, though, is that you don't want to really see a dramatic difference between the 2 groups. So these vaccines were monitored for at least 8 weeks of safety data after the second dose, and it's really unusual to see adverse effects beyond the 8 weeks. It's really, and for people who've had the vaccine, they'll ask you to stay nearby for like at least 30 minutes, and that's really the time when most adverse allergic reaction events happen. So and the instruction that people are given after they have that vaccine is if anything happens, they should immediately call 911. And people who are known to go into things like anaphylactic shock and who carry an EpiPen, you should always have that with you when you're going for any kind of a vaccine. So 2 of these vaccines have in December and early January got through the FDA emergency use authorization. Both of the vaccines have 2-dose regimens, a little bit different in the dosing periods apart. But overall, they both met the same safety standards, which are the same for a regular vaccine development. The reported side effects so far have only been really associated with that anaphylactic response. So roughly now, there's a report of about 2.1 cases per million for the Moderna vaccine and 6.2 cases per million for the Pfizer vaccine. And again, data on these will be continued to be collected throughout immunizations throughout the United States. So again, just wanted to reiterate that, all the authorization the vaccines go through, and now there are a couple other kind of vaccines. coming through, and they will go through that same emergency process, and they'll all be monitored afterward. There's actually quite an extensive system for monitoring vaccines in the United States, and they have to be reported to one of these systems, and there's active surveillance going on about the vaccine trials. So again, the FDA is monitoring these very closely. So I said there's a couple of vaccines on the way. Moderna and Pfizer have gotten through the EUA process. There's also AstraZeneca, which is one of those viral vector-based vaccines. The same thing with the Sputnik 5, which is coming from Russia. All of them have 2-dose regimens and different types of effectivity. Although I want to point out that these 3 vaccines, their clinical trials occurred fairly early on before the variants that are now popping up, which may be related to why this one, which was carried out in the UK predominantly, may be appearing less effective. Another vaccine that's coming up is one from a company called Novavax, and that will be one of those vaccines that's a particle vaccine that's non-infectious. So just a couple of facts about what happens after, or who should get vaccinated. So even people who have had COVID-19 should get vaccinated. And part of that is because you want to make sure that you really get your body ready to respond so that you can avoid a severe case of the disease. There was just a study published this Monday that provided evidence that those people who actually had an infection may only require 1 dose, because what they are seeing in people who had COVID and got the 2-dose regimen is that they experienced very painful side effects on that 2nd dose. So that's something that more research needs to confirm, but everything around SARS-CoV-2 and COVID-19 is changing every day as new data comes out. So it's just something that we have to keep looking at and making sure that we can take all the data and turn it into actionable practice. Another question people have is whether this will cause you to get a positive COVID test. And because the vaccines are not derived from live virus and cannot create an infection, you will not be positive on a molecular test. What will be positive and what should be positive now is your antibody response. So there are some situations where people who are getting the vaccine might be called back later for an antibody titer. Depends on what program and where you're getting your vaccine. And the COVID, mRNA vaccines will not give you COVID. None of the vaccines that I showed in that previous slide are capable of actually giving you COVID-19. So you can, though, in the in-between time, in between doses, as you saw from some of the data from the Pfizer trial, you can become infected in the course of your vaccination. So that is a possibility. I wanted to say a couple words about the variants that have been popping out. So as I'm sure most of us are aware, creating genetic variants with viruses is common. That's why nobody really builds immunity to the common cold, or influenza is another virus that changes so rapidly it's difficult to get the vaccine made in time to respond to it. So it's looking like SARS-CoV-2 will be somewhat similar to the influenza story, where the ability of the virus to generate variants is so rapid that we might be looking at having to constantly modify the vaccines to keep people protected. So again, I think, we know that we're able to prevent severe infection, and that's really worthy and a good enough reason to get the vaccine. All of those variants, as they're coming out, are now being monitored. There's been very large surveillance networks put into place. So here's a couple of them. Nationally, we have a surveillance system in place that's really coming from connection of all the health departments in all the different states and the public health system in the United States, where they are pulling samples randomly from people and sequencing virus to keep up with that. The commercial diagnostic laboratories, as well as the vaccine companies themselves, and another consortium led by the Centers for Disease Control. So there's a lot of effort out there to monitor the new genetic variants as they arise, not only in the United States, but actually throughout the world. So just a reminder that, vaccination is only one measure to help stop the pandemic. So even when we're all vaccinated, I think, until we get to a place where we have very good vaccine coverage, we're still going to have to do things like wear a mask and keep social distance and clean and disinfect and wash our hands, because as the new variants arise, we want to make sure we are vigilant and keep ourselves protected. But vaccination is a safe way to build protection, especially from those severe illness. So using both natural immunity and immunity generated by a vaccine will be really important to get us back to something that looks like normal. And that's all I had tonight. I have the CDC website here. They're a really great source of information. There's a lot of misinformation out there on the internet. So it's really important to get it from a source that's reputable. And I have my email here, from my part-time job as a trustee at Tuckahoe, and wanted to encourage anybody to reach out. And if they have any questions later, we're going to have some question and answer right now, but certainly you can email me. And as always, as you're going through the process of being vaccinated, consult with your doctor. Thank you.
Thank you very much, Renee. To be fair to everyone, I'm going to kind of read the question to Renee.
Sure.
I will start it from the email sent in advance before the presentation, and then we will go through on the chat box. Sure. And then later we will ask people to raise their hand, and they can unmute themselves to ask questions. So the first question I got is from Andrea. She said that she is in a quandary about getting the vaccine because her hypersensitive nature and reactions to vaccines and chemicals. But she works in the school, so she knows she's at risk, but doesn't really know what to do. Her initial thought was to get some blood work done to make sure her metal levels are low and that she don't need to detox her systems. And any thoughts are welcome.
Yeah, I think anybody who knows that they have, some kind of hypersensitivity to anything, so that's all your immune system, you definitely should speak with your, a medical doctor about getting the vaccine. So, again, even those people who go into an incredibly dangerous situation like anaphylactic shock, if they have an EpiPen with them, it's resolved, very quickly. So it is safe for people, who have some types of sensitivity to get vaccinated, but you really need to consult with your doctor to make sure that you have what you need when you're being vaccinated.
Okay, the next question will be from Mary. Can you go back and show the slides with the RNA and DNA and the engineering and allergen?
I sure can.
Thank you.
So just to show the slides, or is there a specific question?
Mary, would you like to unmute yourself and ask the question if you have?
Yes, hi. Just showing the slide would be great. Thank you.
Yep, no
And
problem.
yeah, if you want to give a quick explanation again, if you don't mind.
Yeah, this is just a basic, cell.
And the one after that and the slide after that you showed. Yep.
Sure. So this was just to show basically, how information in DNA is encoded into proteins, which make all living things. So in the nucleus, which is a structure inside our cells, which kind of serves as the brain of a cell, there's DNA, and that contains all of, basically the blueprints for everything that we need to make a human. Because DNA doesn't leave the nucleus, it has to get the message out into the outside, the cytoplasm of a cell, where all of the enzymes are that can make proteins. And to do that, DNA is transcribed into what's called messenger RNA. And it really is a message that then moves out of the cytoplasm, out of the nucleus, into the cytoplasm, and is, can be encoded into protein. The other reason why messenger RNA, you don't want it to stay around, is this is how a cell controls expression. So if you say, oh, I need, I'm in a stress right now and I need, chemical X to serve to balance that stress, part of your DNA will open up, produce a message RNA, go to produce a protein. As that protein is released and say, a stress response goes down, then this RNA production will stop because you don't need it anymore. So this system, is also used to regulate the production of proteins in the body. And that's really mediated through RNA. So RNA is not a stable chemical because it has to be able to go away when it's not needed anymore. So I think the next slide is, my little analogy where, I like to think of DNA as our blueprint. It tells you how you're going to build whatever organism is made up of it. So if you're a human, it has all the blueprints for a human. And at mRNA, you can think about that as the engineers and the construction people who are able to read and translate this message into an actual product like the buildings. So here the buildings would be represent proteins.
Okay.
Perfect. Thank you.
Yep.
And also Mary comments very much appreciate the presentation has gone into this, the preparation that has gone into this presentation, very informative. And the next is also a comment from Adam, great presentation, thanks Renee. And the next question is come from Nancy. She commented on great presentation. Is this vaccine grown on an egg?
No. That actually practice of growing things in eggs has stopped because it elicited a lot of allergic reactions. So these vaccines are synthetic, totally, derived outside of any kind of a living system.
Okay. All right. So the next question is from Julia. Wonderful presentation. Has anyone vaccine died from anaphylaxis related to the vaccine?
So of the ones that have been reported so far, no. But, I think because most people who know that they're prone to that carry an EpiPen with them.
Okay. The next comment also from Suzanne Savina, great presentation. And the question from Janet. Does the antibody test indicate that you have had COVID-19 or just a strain of the COVID virus? I believe that this information has not been clear when mentioned.
Yeah, so there are a couple types of antibody tests. So when you get vaccinated, as I said, all of the vaccines are directed toward that spike protein of the virus. So if you get an antibody test that tests for that same spike or antibodies to the spike protein, You don't know if it's because of the vaccine or if it's because of a natural infection. But there are antibody tests that test, I'm going to show you here in this slide, that test for proteins that are inside here, which are nucleocapsids, which protect the viral RNA. So some antibody or some tests will pick up antibodies to those proteins. If you have those proteins, you had a natural infection. So there are different types of antibody tests which can tell you where your antibody response is from, whether it was from a natural infection.
If your antibody test is positive, it doesn't necessarily mean that you've had COVID-19. It just means that you've had some type of COVID, because there are other COVIDs.
No, the antibodies are the antibody tests are specific for COVID-19, for the SARS-CoV-2 virus.
Okay.
It won't pick up the MERS virus. It won't pick up SARS-CoV-1.
Okay.
Yeah, and again, you know, if you go look on the FDA website at their emergency use authorization for whatever antibody test you're going to get, it will show you cross-reactivity to different kinds of viruses, and those you know, COVID-2, MERS, similar coronaviruses are every antibody test is tested against those viruses.
Okay. And also a comment from Linda. Linda said, great presentation, very important information. Thank you very much. And also Ginger, I'm not sure about your question. Would you like to ask yourself? You can unmute yourself.
It was Donald. He's going to do it.
The second shot, is it stronger than the first shot?
I don't think the dose is any different. I'm not sure, Donald. You know, sometimes they do that in this trial. There must have been something in the early data that told them they needed to do 2 injections to get the immune response to the point where they needed it to be.
So I'm assuming that it's cumulative.
Yeah, you'll get a much stronger response on the second injection. Now there's, I think J&J is coming out with a single dose vaccine.
Right.
How do they determine like one is 3 weeks apart, one is 4 weeks apart?
So that's usually in some of the preclinical work. They just are, you know, they'll do testing to see when they get the best response, you know, how far they have to space those 2 doses to get the best response.
Thank you.
Okay, the next question is from B. Dr. Fauci stated that triple mask should be worn. What are your thoughts about this?
So, you know, there's all kinds of thinking behind the masks, but I think what's been shown with data is that if you have like an N95 mask or a medical grade mask, that you have a fairly good level of protection. Of course, the more masks the more masks and the higher quality masks you wear, the less you're going to transmit virus. And really, I want to remind people, you know, you wear masks in case you're sick, you don't want to spread it. So it's really to contain things more than it is to protect you.
Okay.
But a lot of times with this virus, you don't know when you're infected. So it's very important to wear a mask.
And also the next question from Caroline Tennant. Caroline, would you like to ask for yourself? Caroline, you still here?
Yes, I'm still here.
Okay.
My question is, you know, with the Johnson & Johnson coming down the pipe for authorization, how does it differ from the Moderna and Pfizer? And how do you compare that, like one shot as opposed to 2, and will it protect people from underlying medical conditions?
So it is a one-shot vaccine, and they are reporting some of their clinical trial data already, and it looks very effective. Again, they're doing their trials while these variants are around. So it looks a little bit less specific and sensitive. So, but still protective, from severe infection. It's not an RNA vaccine. The J&J vaccine is another type of vaccine. It's one of those virus engineered. So it's actually the type where you take the nucleic acid, the RNA from the SARS-CoV-2 virus, and put it into a viral vector, like a common cold virus. That's the structure of that vaccine.
Okay.
So that to me sounds like it may work better, like a flu shot, right?
Yeah, right. So it'll be one shot, and it'll be certainly easier and faster to get people vaccinated. So we'll wait to see it move through its approval process.
Okay, thank you.
Mhm.
Great presentation.
Thank you.
The next question is also from B. If most people are wearing masks and supposed to prevent one's infections, why are so many people getting sick?
Well, I assume that maybe people aren't wearing masks and people aren't socially distancing and following all of that guidance. I mean, I think we can see, there are times when there are events that have been held, and immediately after that, people who, were seen not wearing masks were sick. So I think that really comes from people not following the guidance.
Yeah. And also the next question is for me. When will the recording will be available? I think it probably will be available sometime next week, the latest. And from Charlotte, receiving vaccine on February 4th, but having surgery on February 9th. Any problem with that?
Again, I think that's something you should review with your doctor, but I think it would be, probably low risk.
Okay. And the next question from Anne-Marie. I had no reaction to either of the two vaccines. How do I know if I had an immune response?
So you have to get an antibody test. The antibody test will tell you.
Okay.
So you need to get an antibody test toward the spike protein.
And just so everyone knows, we will be offering the antibody test on Saturday at the Shiloh Baptist Church in the village.
Thank you, Omara.
Do we know which one it is, Omara?
Which of the tests? No, I don't know off the top of my head.
Manufacturer. So because that would be important. There will be some that won't pick up a vaccine response, but we can find that out, right, Omara?
Yep.
Okay.
And the next question is from Ginger. What is the cost of developing a vaccine?
The cost of developing a vaccine?
Yeah.
Millions and millions of dollars. So I can tell you, the cost of I'm trying to think of a therapeutic, can be like hundreds of millions of dollars. I mean, it's a huge undertaking. It's very expensive. It's one of the reasons given that drugs and vaccines are so expensive.
Okay. And the next question, after getting the vaccine, is it a fact that you cannot contract the virus?
No. Right. You saw that in some of the data that came from the vaccine trials. People did catch COVID. Right now, we don't know if that was because they came into COVID before their immune system had responded from the vaccine, or if it's, some of that was caused by the variants. So you can still get infected while even though you've been vaccinated.
Okay. And the next one will be from Eva. Eva, would you like to unmute yourself and ask the questions? Eva, is still here? Let me see if she's still here. Looking for Eva. Okay.
Am I unmuted?
No, you are okay. We can hear you now.
Okay.
Okay.
Thank you very much for all the information. I have a question based on a personal experience. 20 years ago when I had cancer, I needed an IV contrast injection. We were trying to figure out if I had any metastases. And during the test, I developed a very really serious systemic reaction, allergic reaction. So this is a personal question. Could you tell me whether you heard about it, that the IV contrast material can cause, act like a allergic material and would lead to problems with the shot, with the vaccine?
You know, there has been none reported that I'm aware, but again, this is
Right,
something
right.
that I would ask your doctor. I would be more concerned about the fact that you had a severe reaction to something. So to me, that puts you into a category of people who should be watched if they receive the vaccine. And you know, so you should definitely let your current doctor know that you had that reaction if you haven't,
Right.
and make sure the people who are giving you the vaccine know that you have that potential.
Right. I always tell them about every kind of procedure.
That's
I'm
good.
sure what they know.
Good. Keep doing that.
And the second question is that I did not hear it correctly. When we wear our mask, is it, I think you said that it helps a lot to spreading, but I'm not spreading. So in my case, versus the protection. So I didn't get it, which is what the mask does better, protecting me who doesn't have the virus, or if I have it, I would spread it more or less. So that's not clear to me.
Yeah. So really, you know, you want people to wear a mask so that if somebody sneezes or coughs or has that kind of projection, that a mask really will prevent a lot of that from going through. So
Yes,
that's the main reason, you know, so
Yes.
if somebody coughs and they don't know they're infected,
Oh,
you
yes.
want them to wear a mask. So it's not only people who have the virus, everybody has to do it for it to be effective, but it's really what the main reason you want that is so when somebody coughs or sneezes and they happen to be infected, they don't.
And they don't
spread it.
know.
Yeah,
Right, right. Thank you very much. It's
you're
very
welcome.
informative.
Thank you.
Any more questions?
I think there is one more from Maurice.
Okay. Hi, Maurice. Would you like to ask your questions?
Oh, sure. I was just wondering, I've heard somewhere that even after I will take the vaccine in both doses, I have to stay away from my children and grandchildren. Why is that?
So I think, you know, you don't have to stay away from them, but you have to remember that you can still get sick and you can still spread the virus. So that's why I said, even when we get vaccinated, we'll still have to wear masks and we'll still have to have the hand washing and social distancing until we can get enough of the population vaccinated that, you know, we can really, really slow the spread. So eventually we'll get there, but until the majority of the population is vaccinated, we are still at risk for spreading the disease.
But even though I have young grandchildren who do not qualify to take the vaccine now, because the youngest who lives in New York is 14, so which means that he's definitely not qualified.
Right, and so they're doing clinical trials on children now, you know, so that will come. But you're right, there's no vaccine approved for people under the age of 16.
Yeah.
So until, it'll get better as the adult population is vaccinated, but until we can really vaccinate everybody, we still have to keep the precautions in place for spreading infection.
I have to go back to my original question. Once I have the vaccine, even though I know that I don't have it because I'm keeping myself away as much as possible.
Mm-hmm.
There's a possibility that I could catch the virus if I go out and visit. But I mean, even with a mask, visiting, if I go to my children's home, my son's home.
So if you are not wearing a mask, or even, you have to keep all of those protections in place. Wearing a mask, social distancing, washing your hands. If you don't, if we don't do those things, you can get infected.
So what really I'm saying is that we cannot have a meal at our child's house yet.
Yeah, unless you're, washing your hands, wearing a mask, and when you take your mask off, you're sitting 6 feet away from everyone.
Okay, all right. Thank you so much.
You're very welcome.
The next question maybe is for Omara, because I'm not familiar with the church. So the church's name is Shiloh's Baptist Church. Will give them a vaccine on Saturday.
That's correct. From 9 a.m., not the vaccine, we're doing testing.
Yes.
So there's the PCR test, the nasal swab, and the antibody test from 9 a.m. to 2 p.m. And you can just walk in at any time within that time frame.
Yeah, we should give a shout out here to Trustee Andino. Since the pandemic hit, she has been able to arrange for testing to be available in the village. And that's a pretty important public service to provide. So thank you for doing that, Omira.
No,
I have a question, Heidi. It's Ginger. For Renee?
Okay.
Having had the first vaccination, is it worth going to get the test for the antibodies to see if I have any in my system thus far? I go for the second one next week.
No, just wait till the second. You should wait at least, 2 weeks after the second vaccine.
Okay, thank you.
Mm-hmm.
I see someone is raising the hand, but the hand disappeared. So your question was answered, right? Okay. Anyone else have any more questions?
I have a question, if I may. I'm hearing impaired, and the math thing is really hard for me because of the speed reading. I was just wondering what your thought is, how long that will continue. Is it going to be when we get herd immunity?
So I'm not sure I got the whole question.
Okay. I'm hearing impaired, so the math really is difficult for communication for me.
Yes.
And I was just curious how long the math thing will continue.
And until we reach that herd immunity, right? So
Yeah.
we don't have vaccines yet for kids, and that's probably we're going to have vaccines. We're going to have children vaccinated before we can reach that herd immunity.
So you're saying another 2 years?
I would say at least another year.
Okay.
Then I have a question. I have heard people have this transparent mask. Will that work?
Ah, that's right. Yeah.
Yeah. So if Carolyn, if you can get those transparent masks when you have to have like certain people you want to communicate with them, maybe they can wear that kind of mask. That might help for you to communicate.
Yeah, thank you, but it's not for me. It's for everyone else.
Yeah, I know. But when you have like essential like relatives or friends that you communicate regularly, yeah, that's what I mean. Yeah. Okay. Anyone else? Questions? It's almost 8 o'clock, I think.
Yep.
Elaine, I have one question. My understanding from listening to Dr. Fauci is that once you're vaccinated, and if you contract COVID-19, it's unclear whether it's the level of transmissibility. you know, it may be less than someone who isn't vaccinated, the level of infection that you have. So the
Right.
question is, and he indicated that was being studied. When will we know that? Because once we know that, it's a whole different story, knowing if we are carriers or transmitters.
Right. So, you know, the hope is that if you have a vaccine, you don't get a severe infection, you don't carry the viral load, right? So that the virus doesn't make a lot of itself, and that contributes to the ability to spread it. So, you know, realistically, I think it's going to take, you know, at least 6 months to a year before we have enough data to make a firm statement on that.
Okay, thank you.
Mhm.
So I'm going to say the last question, and then if you have any further question, as Renee said, that you can either send it to her email address, or you can send it to the library. We can forward it to her. That's all. That's okay too. The last question will be any definitive word about local pharmacies getting the vaccine, and also mask with filter okay to wear. Mask with filter.
Yeah, I think the N95 masks have filters in them. So yeah, any kind of layering that you can add to that helps. So there was a couple things I saw come out in the news today where in the state of New York, they have additional sites they are setting up to do vaccine distribution. And they're also working with some pharmacies like CVS, Walmart, et cetera. And they're starting to post the names of those. So I think, you know, you just have to really keep looking at the news, keep looking at the New York State Department of Health website as they put more sites, you know, onto the system to vaccinate people.
I saw that the CVS in Eastchester is going to have it.
There you go. I mean, I've been wondering because they put that little booth out back. Is that what's part
I
of
don't
that?
know. I happened to see it on the patch for the online newspaper.
Yeah, okay.
It mentioned this, it mentioned the
I
specific.
think
Yes.
that
We'll be getting it.
Right.
Great presentation. Thank you, Renee.
Yeah.
Thank you all for attending.
So the last things I want to say is that Omara just want to make sure that the Shiraz Baptist Church will offer antibody and PCR made available on Saturday, not vaccines. So just want to make sure of that. And thank you everyone for joining us. And thank you so much for Renee to give us this like very thorough, very informative presentation. I hope the presentation do give you a lot of idea, like instead of scrolling around the internet to find information. I feel like I hope this presentation clear out some of your doubts and your concerns. So thank you very much for everyone. And the last things I want to mention is that we are going to have because February is the Valentine's February is also the Love Your Library month at the Tuckahoe Public Library. We want to hear from you. We are going to have some plan coming out, and we will send to everyone to let everyone know. If you haven't signed up for our newsletter, please sign up to receive our e-newsletter. This is the way that you can get all the information, all the updated community news, and the library news and the library events from us. Thank you very much, everyone.
Thank you.
Thank you so much, Renee and Kaiti. That was wonderful presentation, Renee. You really worked very hard and very informative. Thank you. A round of applause for her.
Thank you.
Thank you.
Thank you so much.
All right, everyone. Stay safe.
Have a great evening, everyone.
Thank you. Thank you.
Bye-bye.
Bye-bye.
Bye, Renee.
Bye.
Bye.